Well, why don't we get started? Let me start off by thanking the speakers again for their excellent presentations and just a wonderful set of talks related to translational neuroscience, capped off by Josh's comments related to animal models and also the policy comments. Just a terrific group of people. I think we want to encourage folks to ask questions on the chat line, and also begin a discussion amongst the presenters as well. So I think the first place to start is to invite the presenters to maybe see if they have questions of each other or particular thoughts that came up in relation to the different talks. If we were all in person, we would have had more of a rich discussion with the presenters in vivo, but this is an opportunity to let them interact a bit as well.
Ned, I can start off if you like. I'll just add a comment relative to Josh's comments about use of animals to either address hypotheses or proof of concept studies. So just as an example, so what I presented today, I hope, are a set of robust findings that I interpreted in a story of cause, consequence, and compensation. But in fact, all I showed you were data that were associated and did not show you anything that actually proved the linkages among those. So to address one of the things that we're doing now in macaque monkey prefrontal cortex is using a viral mediated genetic expression of a gene that essentially silences the activity of pyramidal cells, and an expression of an epigenetic probe in parvalbumin cells, so that we can, in a slice preparation from those monkeys, actually test the hypothesis that I suggested was a compensation, which is, are the parvalbumin cell inputs to a pyramidal cell that is quiescent, artificially quiescent, downregulated relative to the PV basket cell inputs to a pyramidal cell that was normally active. And so to me, that's an important use of an animal to do a proof of concept test of a hypothesis in humans that we can't test in humans. And I think, in fact, someone's comment that I think it was Richie, the interplay between animals and human studies is critical to advance our actual understanding of the disease process. So I just wanted to throw that in as an example of the types of things that Josh has mentioned.
Josh, you are muted.
Let me check my settings.
Now we can hear you.
We can hear you now.
Okay. All right. Sorry about that. I think maybe we did the double mute. I was just say that formulating that hypothesis is important, not just to be able to properly interpret the experiments, but also to justify the use of an animal, right? Particularly an animal like a macaque, where the expense and the is considerable, and you want to try to minimize the use of the animal, so you're not just engaging in a fishing expedition to just look for the effects, right? You have a specific hypothesis, and I think that's important both from the perspective of the kinds of experiments you're describing, but also those that Guoping is working on, right? You want to go into you want to go into the animal with either a specific hypothesis or the knowledge that you tried in a lower animal and couldn't find what you were looking for.
I have a question for maybe for Josh mostly. From the we have since unlike Ned, who is had been in the monkey business for a long time. I'm new, but I realized that in the last few years, because of the technology innovation, there are a lot of young people really interested in doing a postdoc and training to working with the monkey models. The problem they always worry is that, once they get a job, and we don't have enough trained people actually to take the tasks, and they're very worried about how long it will take for them to get the data, to get a tenure. So I'm just wondering whether there are a lot of special fellowships to bridge them in autism research, in all other things. Is there NIH has done a great job to have specialized IFAs for marmoset and other things, including brain initiative. Is there any thinking to help with the young people, make them there's a bridge fellowships or grants specifically for these long-term studies or slow models that help them to calm them down. They think that they are willing to take the risk with some support from NIH, specific bridge fellowship. Is there anything like that is ongoing or thinking?
There was some thought to those kinds of things, especially with the Brain Initiative, but no action yet.
I guess I have more of a generic comment. So we've done a little bit of primate work, non-human primate work with Kathy Grant over the years. And, really there's a lot of similarities with what we study, but from our standpoint, or from my standpoint, one of the really interesting things is that we can really treat them as individuals. And for a lot of rodent work, and this is, I'm mostly talking about what I do, we tend to ignore the genetic heterogeneity and diversity that is actually really critical for understanding and generalizing some of the work we do. So, I think this is just more a self-reflection than anything, is that, a lot of work, the modern neuroscience systems work is done in C57 black 6 mice, and maybe stress people do it in, C3H or some other line. I do think that there's a lot of value to be gained in kind of looking a little bit more, using some of these same tools, but looking at how does genetic diversity kind of impact this. So it's not, we can get some idea about individual variants versus, not within the same line, but true genetic variance and variability. So I think this is, when Guoping mentioned how each monkey is an individual, it really, it kind of highlighted the fact that, this is really, to me, a great strength of non-human primate work, is that you're not, narrowing in on a real kind of clonal population. So I think it is something that, I hope, I know I can do better here, obviously. So it's self-critique, but I think this would benefit it would benefit from a lot of the approaches we use to kind of look more broadly and make sure we're not just chasing down a phenotype that we see in C57 black 6 mice. So I think it's a little bit of a riff off the idea of we can learn a lot from non-human primates, but I think we also have some tools to investigate diversity within, or genetic diversity rather, within other preclinical animal models that are maybe a little bit faster than macaque or marmoset.
Well, I think the question by the student about understanding the molecular implications, I think when you get down to the molecular and cellular questions, you can really nicely do the genetic diversity stuff in humans, right? Because you can do it in cells. And these organoids like the Broad is developing that combine cells from many, many individuals. So you get genetic diversity in not just in the same dish, but in the same ball of wax. I think those are also approaches, but obviously they're only useful for certain kinds of questions.
I think just a comment on the non-human primate stuff. I think the points have been made that the evolutionarily sort of conserved circuitry and molecules, are important from the standpoint of translating from rodents to non-human primates to humans. And there's a place and time for all of these different species and models that Josh was highlighting. The individual difference question is a really important one, and you certainly see, you can see the individual differences in non-human primates, but there's also the tension around having sample sizes that are sufficient to feel confident in the findings. And some of as you know, many of the non-human primate studies are limited from the standpoint of sample size due to resources and cost and all that. So it's tricky, but Guoping, I also think that thinking through special training programs to help people exploit and learn about non-human primate models in relation to rodent models and human work could be an interesting pathway that we could think about trying to facilitate.
Yeah, I think this is a really good point, and especially on the, there are many things we need to learn. But one thing I'm worried about the marmoset, we, NIH initiated a lot of support of, program for marmoset. But right now, all the marmoset, we just had a meeting today. All the marmoset are individual colonized. Each individual colony are unique. We did a whole genome sequence, compare them. Some are very, very homogeneous. Some have, some are better. But just as, Tom said, that if we don't, what do you call them, generalize them or, interbreed them, then your study behaviors are based on one genetic background in Wisconsin, non-human primate center, and one is Southwest, one is at MIT. So there's a very little comparison. So I think it's how do we design a program to analyze all their origins and all this, and then share the sperms to outbred them to make sure we have something that okay. Otherwise, basically Wisconsin has their own C57 marmoset and we have our own. So it becomes a real problem. I'm very worried because we should solve this problem at the beginning now. Later will be too, if we don't solve now, it will be too late to change things.
Well, I mean, we have the tools because we know the genetic makeup of most of the animals in the different colonies. So that's a good place to think about starting. Perhaps we should take a look at some of the questions that have come up through the chat, Richie. I'm just going to pull that up.
Yeah, there are a bunch of specific questions that didn't get answered earlier, and they may lead to other issues. So here's a question for Guoping. It's a question about the Shang-3 monkeys who and whether they show overall hyperactivity and also whether they show any unusual EEG activity.
Yeah, that's a very good question. So they don't show overall hyperactivity. They actually show lower activity if you don't. It's just monitor the 7 days a week, 24 hours a day to monitor them. However, they are very they show high reactivity when someone walk by or you show them a video. We do social eye tracking. You show them a video, they will screaming and jumping. Unlike wild type, they will look at a video. They will look, but they will be very hyperactive or hyper reactive to whatever the new things you show them. So even a person walk by will cause that a problem. And so the second part of the question, sorry,
what was about their EEG activity?
Yeah. So we are still doing the EEG. We have not made a conclusion because originally we only had 3 founders. Now we are working on this about 20 F1s. So we're doing EEG now. We don't have a conclusion yet.
Okay. Thank you. Let's see. We'll sample these. This is a question that came up in Tracy's in response to Tracy's talk. If an individual recovers from trauma, how would that reflect on the zygotes?
The individual recovers from trauma. I mean, this is something we talked about a little bit before my computer crashed, I think.
Yeah.
Which is understanding the signals that relate to stress or trauma versus those that are more related to resiliency versus the complexity of the lifespan. I think Josh alluded to that in his NIMH overview, which is, and I think as I logged back in, I heard somebody, maybe Tom, talking about that oftentimes in animal models, we're studying, a healthy animal in a healthy environment with healthy food and healthy bedding, and what really are we modeling? And I think that kind of leads to that point of trying, how do we understand a complex background with contributions from mom and dad, and then a complex gestational experience that all feeds into disease risk and resilience. I think that we do need to become more complex neuroscientists. And I think Josh had something to that effect with modeling. I think we really need to head in the direction of understanding modeling rather than this heat map and that heat map. Like, how do they intersect and what can that inform us toward translational outcomes? I think it's going to be the next decade where we really start to model that data.
Yeah, that's great. Thank you. This is a question which I think was posed to several of you for the general discussion. So the question is this: studies using functional MRI have suggested that increased amygdala BNST activation relates to heightened anxiety. Oh no, this is actually a question for Tom. So it's for Tom, but others may chime in too. Studies using functional MRI have suggested that increased amygdala BNST activation relates to heightened anxiety, whereas your work has shown that distinct pathways, cell populations may exhibit different patterns. What do you view as fruitful avenues of merging molecular and cellular research, which is very high resolution, and systems neuroscience / neuroimaging work, which has relatively low resolution? And this person also asked about recent developments using more granular methods like multivoxel pattern analysis in human imaging work.
It's a great question. I don't think I can speak to multivoxel analysis in human imaging with any authority or actually any knowledge. But I do think that there is an interesting, the interesting greater point is when we, if we look at human MRI, resting state MRI, we see changes in connectivity. But if we zoom down, we see these, there's a very, there's a host of different events happening. There's different inputs. There's GABA, there's glutamate, and there is this big bridge between the really finely detailed, Josh will refer to the single unit work as this really finely detailed work in vivo, and it absolutely is. I'd say you can even get further levels of detail with molecular analysis and looking at different aspects of synaptic transmission. So how do you scale that effectively? How do you let this molecular work inform some of the imaging work? And this is something that I think is there's some ways to get there. I think part of it is, we've spent a fair amount of time trying to do whole brain sort of activity mapping to generate, using iDisco and ClearMap to generate light sheet microscopy to generate maps of, snapshots of activity in the brain at specific time points. We had a grand idea at one point, we were going to do that and then do resting state MRI and then, develop a Rosetta Stone for transitioning between the two of them. That was pretty difficult, I think, would be the first word to come up. So it's hard to really do it on that level. I think we've moved some things forward, but part of it might be, if we have really discrete molecular manipulations that can have robust effects on circuit dynamics and robust effects on behavior on the level that molecular systems neuroscientists are looking, in my mind, it makes sense to try and scale those up. I mean, we can do rodent fMRI. Guoping showed some amazing data with Schenk. You can engineer monkeys. I know Kathy Grant has been doing and Ned as well has been doing dread work in monkeys. So, I think we're at the point where we can apply some of these circuit methods we use in rodents that give us fine-grained molecular perturbations and look at how that might impact some of these larger scale, different dynamic processes with MRI. So I think that to me is a path forward. I mean, my very first science exposure was at Brookhaven Labs doing PET, not even PET imaging, like kind of related PET work in the medical department there. So I have a soft spot for, application of new molecular probes to study neuronal dynamics of other systems. There's a great scientist whose name is eluding me right now, who has developed a bunch of really exciting molecular probes for magnetic MRI, sort of molecular probes for looking at different aspects of signaling in the context of a whole animal. So I feel like we're at the point where we can bridge those a little bit better. We have really discrete biosensors where we can look at norepinephrine in the PFC and relate that to pupil response. And then maybe I know Ian Shee at UNC has been integrating fiber photometry with biosensors into MRI analyses as well. So you can start to design computational models of how these various signals interact. So I think it's not easy. It takes a lot of different skill sets. As Josh and many others have argued, the need for really rigorous computational approaches becomes more evident when you talk about moving between these different levels. But I think, my gut tells me that with enough resolve, we can get there. And theoretically, the Brain Initiative is investing heavily in these ideas. So I think that we're getting closer to bridging some of these molecular circuit systems that are, so rapidly developed and applied in rodents into defining a better understanding of some of these broader signals we can also measure in people. So I think that's my take. I'm sure there's other better, more nuanced takes from the other panelists on this, though.
Yeah. Do any others want to comment on this? If not, I wanted to, I appreciate that comment. It's very helpful, Tom. I wanted to ask a question about another cross-level integration, and this is really, I think, mostly directed to Josh. Most clinicians who are applying behavioral treatments to psychopathology do so, and if we look at evidence-based behavioral treatments, virtually all the treatments that are currently in use today are not treatments based on anything we know about the brain. And I wonder if if anyone, and particularly Josh, has thoughts about thinking about neurally inspired behavioral interventions. So behavioral interventions that are specifically designed based upon what we know about the brain. There's a little bit of work on memory reconsolidation and those strategies applied to trauma that I think is a good example of this, but there are preciously few, I think, behavioral interventions that truly have emerged from an understanding of brain mechanisms. So I'm be curious to hear your thoughts about that and whether you think there's a role for investing more in that area.
Well, there's a few different answers I could give for that one, Richie, and thanks for bringing it up. The first is that you're right in the sense that a lot of the behavioral interventions actually have informed theories of how the brain works more than theories about the brain work have informed changes in treatments, although you cite one. And There's a couple of areas, though, where I think progress is being made. One is not exactly what you're talking about, but in terms of neurofeedback. So there's a number of folks who are out there, putting EEG or MRI, connecting that up with various kinds of behavioral or cognitive therapies or even just with, sit in the scanner and try to turn down your amygdala. But let's say that class of neurally informed approaches that measures neural activity as a guide for whether or not things are headed in the right direction. I think that's one thing people are exploring. The second area goes back to what Tom just said about computational approaches and what I alluded to in terms of, for example, reward learning. There are definitely people out there who are testing not really yet neurally informed models of cognitive therapies, but computationally informed behavioral models. For example, to use reward learning paradigms and reinforcement learning models to try to repair reinforcement learning deficits in individuals with depression. Mm-hmm. So we've got someone at NIMH who's doing that in kids, the various folks who are doing cognitive remediation therapies like Sofia Vinograda for schizophrenia that are really trying to get at some sort of universal underlying deficit. David mentioned the notion that maybe working memory is some underlying deficit that leads to lots of different symptoms in schizophrenia, and I think that at least partially underlies Sophia's work. So there are some attempts to begin in that area, and I think as our ability to understand and translate neurobiologic theories from animals into humans and monitor them, I think the chances of being able to develop more focused therapies that are neurobiologically based is intriguing. Whether those will actually generalize and be helpful in the way that many psychotherapies are is an open question.
No, that's great. Thank you so much. That was very informative. There are a bunch of questions coming in now. There's a question from Margot that is really appropriate for each of you to address, I think. The question is, how have your research programs changed as a result of the pandemic? In what ways were they negatively impacted, and what practices will you bring forward to the hypothetical pandemic-free world of the future?
Richie, I'm happy to start. I think the pandemic has had a huge trainees. I think that many of them live alone and that they're at a particularly vulnerable age. The pandemic has been enormously stressful. We've had periods of time where the graduate students weren't allowed on campus. That was very stressful to them. Even when they came back, I have a fairly large lab, and so we had to divide into teams, and they couldn't cross-interact because of contact tracing. We had problems with interactions with the vivarium staff, and it was an enormously stressful period that required a lot of individual meetings to try and keep everybody on track. But Zoom is not the same as meeting in person. It's not the same as having a lab where there's stuff buzzing all the time. And I think, people coming in early in the morning versus people who were coming in late in the afternoon, staying into the evening. I just think it had a huge impact. We are social creatures. So I think that it has had a big influence on their perceptions of how their project was going, of how they feel about science. So I think there's a lot of work to do post-pandemic to get everybody back involved and excited. Doing Zoom, not the same. Yeah, I noticed just the difference, and I took a picture of every one of our lab meetings on Zoom over the course of the pandemic, and you can just see the look on their face over time from, kind of a happy, this is kind of different, to quiet, to now we're sort of I'm concerned about, several members of the lab. So big impact.
Thank you.
Yeah, I want to add is it's particularly difficult I found for postdocs in the lab who are, especially female postdoc who has small kids. It's just very difficult for them because daycare is all closed. That the most difficult part is they just couldn't get a lot of them because of they have to take care of babies and do all these other things. Usually at home, babies like moms better than daddy. Daddy are just not as good at taking care of babies. So that's very difficult for them. So I've several of my postdocs actually postponed the job search, because of all these things. So that's even more difficult than in general. So once daycare and the kindergarten or school open, it will be easier especially for moms, young moms.
Yeah.
I think it's. I know a number of my students are listening in, but which is nice. I think it's particularly difficult, and as leaders of a lab and in our other roles, we really do need to extend beyond our traditional scientific, graduate mentoring, I think, and really think a little bit more about everyone's well-being and how we can facilitate that, given all the restrictions that we're facing, including the own stressors that we're facing ourselves. So it is really challenging, but I do think we have to go the extra mile. And I'm sure Tracy, that we talked earlier about that your concerns and what you're doing. So.
Yeah, I think we're looking for a pretty big mental health crisis, as I'm sure Josh is more than aware, coming out of this pandemic for just a lot of communities. Huge impact.
So I'll just chime in. I agree with everything that has been said, and we've seen all that. But I want to respond to the part about how we're going to make things better based upon what we've learned from COVID. And I'll just give two examples. I think so. Now that we've learned, we can do lab meetings via Zoom, and we're much more efficient at it. That's going to make it easier to schedule because not everybody we can do hybrid. And we've always struggled with finding a time of day when everybody could make it because they might have to be someplace else due to clinical responsibilities or childcare responsibilities. So I'm looking at that as a bright side of this, that it will be easier to get everybody together, albeit in a hybrid fashion. The other thing that really was helpful to us is it pushed us to learn how to do more analyses remotely, and so we had to do some investments. So we got an automatic slide loader for a slide scanner, so that now people can put their slides in and go home, and somebody can at 11 o'clock at night put their load in, and nobody has to come in. So these may be small things, but I'm really trying hard to find the positives in this that we wouldn't have learned otherwise, that we weren't pushed to do those things. So I'm hoping that there will be a brighter and more productive side of the future because of what we've been through.
Yeah, I would echo that we've definitely, at some point when we were just not allowed into the lab, definitely took a step back and looked at how we could make better use and analyze our data more deeply. And several postdocs spent a lot of time working on new computational approaches, and so that's a good thing that we'll take forward. I think absolutely flexibility in having hybrid meetings is great. I used to have to leave my departmental seminar early every time because I had to go pick up my son from daycare or my daughter from school. So it was always a little bit tricky. I probably have a slightly different perspective because at the beginning of the pandemic, I had a 2-year-old and a 7-year-old. So I think it was challenging for everyone. It's partially, you have to my take was you have to give people a little bit of grace here. This is a remarkably hard time. And, I have a pretty good, my take is I'm a tenured professor and have a really good deal. And so for the folks who have more ambiguous positions and more ambiguous futures, I'm just, I think is just to recognize that, accept that not everyone had the same situation, and be as supportive as possible. And so that's, really been my take home is trying to see the whole person and understand some of that, and use all the. I hate Zoom more than anyone, one might argue, but it's a great tool. It does, it lets me, if my someone's sick at home, I can stay home and still participate and do things. So if we go back to a situation where we don't have to do that, I'm sure I still will frequently. It's a great way to get new and interesting speakers in. We can have more international participation for some of our conferences and meetings. So I think there are good things. Sometimes it's hard to see those good things when you're walking through the darkness of night. But I think dawn might be upon us. So I think there are some things that we can take from it that are good.
Thank you, Tom. That was great. So we might have time for, there's one other question. This is from Cecile. And this is, I think, a general question that a number of you might address. As we gain more knowledge about brain plasticity, to what extent do you consider neurodevelopment as playing an important role in developing more effective targeted treatment?
Great question. I think that I would say that we absolutely need to be mindful of the trajectory, and that even subtle differences in anything from the rate to the missed steps, give rise to changes in risk. I think, but it's complex. It's not difficult to pinpoint, not easy, to pinpoint, and whether or not you'd want to intervene. And I think this is one of the issues for animal studies, right? We see lots of changes or things that are different, but placing in the context of, is that something you'd want to stop, prevent, or treat is very difficult to know. And that's why translating them back to the human to understand what those changes might mean is going to be really important.
Yeah, I totally agree. I think development is very important, but it's also from treatment point of view, it's a double-edged sword, right? So in adult, you really, in my view, there are plasticity, but as we grow older, the plasticity, the degree of plasticity is reduced. So the earlier, probably in many of neurodevelopment disorder, is better, and that what we think is because small, our brains are constantly try to compensate for whatever the function they are lost or they're losing. So that means that during that time, if you small change may really, help a lot long run and to prevent some of malformations or, miswirings. On the other hand, if there's a side effect or other things, that also can cause, long-term damaging, causing issues. So this is really a very, it's called a critical period. I guess there's a reason. It's a really difficult time to determine. So that's why I think detailed animal studies to ensure that the safety of these things are critical. If we are really treating, treating patients during development.
I think you're going to first see work in development in the more severe end of the spectrum, right? The severe neurodevelopmental disorders that really disrupt function across many, many domains are going to be the ones where the risk is acceptable. And it is harder for me to imagine, intervening in what we know to be neurodevelopmental disorders like schizophrenia, where the outcomes are so much later, and the connection to the illness is harder. I think there, the developmental time periods you're really thinking about are and frankly, that we are already beginning to try to study, which is, in later in adolescence, where you're trying to perhaps harness the plasticity, prevent some of these compensatory things that prevent. Actually, we don't know. We should be presenting or accentuating some of these compensating things that David talked about, but at least studying them. And so I think you absolutely have to take it into account, as Tracy suggested, as Guoping suggested, but it's going to mean different things for different approaches and for different disorders.
I think one of the interesting issues is going to be thinking about how we can evolve and develop treatments that are not particularly invasive, but still can promote resilience and theoretically protect from specific types of psychiatric problems later in life, or at least help protect or buffer. And I think that work, we already have some ideas about that, and that work is going to be based on fundamental understandings of time periods and critical periods, and biology to some extent. But we don't have to be doing invasive things to children to still think about early treatments that could make a difference, even for illnesses that are not severe developmental disorders.
That's really great, Ned, to think about.
I think it's a hugely important point, and there are real opportunities there to begin to explore things. And we know from animal studies that even mild to moderate amounts of stress at particular developmental periods can actually serve to promote resilience later in life. So I think there are some instructive examples.
Yep, you need some grit.
Yeah.
Right.
So I'd just make two comments. One that I think relates to Guoping talk is, so the Simons Foundation, as I understand it, is convening a group to explore whether whole genome sequencing should be done on newborns as routine part of newborn screening to detect kids who are at high risk, genetic risk for autism. And of course, that opens a whole panoply of concerns. But if we're actually going to treat autism before kids become symptomatic, that's the kind of approach that may be needed. And to Josh's point about schizophrenia, I made this point that a deficit in dendritic spines seems to be maybe a key feature of it. We know that those are normally pruned during adolescence, but even though Irv Feinberg 40 years ago postulated that there might be something wrong with that developmental process, we still don't know whether the problem is that during prenatal and the first 2 years of life, there's a failure in the normal development of spines, or whether there's excessive pruning during adolescence, or both. So that is a huge time window that needs to be sorted out to determine if we could identify the at-risk individuals, when is the right time to intervene in a preemptive fashion.
Yeah, great points, David. We've been able to take just a soluble fiber. I mean, if you put mice on, just the horrible environment of what, urban stress and a bad diet, whatever. And it turns out during pregnancy, if you just simply do something like give a soluble fiber in the diet, just increase to produce short chain fatty acids, amazing benefits for resolving these issues for brain development, right? That's really something very easy. Back to what Ned was saying about not a great intervention, just pretty easy to. You can't totally change food deserts in these urban environments, but providing women with soluble fiber, win-win. Perfect for the brain. It's all good.
I'm going to comment on what you think about the prenatal choline administration. the Friedman studies to try to prevent the development of schizophrenia later?
I think you're on mute there, Josh.
About the direction of the effect, no.
Sorry, Josh, you were muted. We missed the first part of what you said.
I don't think I can.
It's this idea that, are there going to be something out there that's like folate?
Right. Actually, there's somebody who keeps applying for grants to study folate because there's clear relationships of folate, but we already know that folate works. So that one thing is not such of interest. I thought with the choline story, there's a lot of controversy as to which direction the result will be in.
I thought the long-term data showed that there wasn't much of an effect on methylation changes in human studies long-term anyway.
Yeah. So anyway, beyond my ken.
Well, I think we're at time. I just want to express my deepest appreciation to all of you for an amazing day and for a set of extraordinary talks. And this is a seminal event for our trainees each year. And I think that despite the limitations of doing this virtually, it has surpassed, I think, all of our hopes and expectations and aspirations. So thank you all so much. And Ned, I'm sorry we're not next to each other to
I know
this, but this
just
is
to make a couple quick comments too. First of all, we should watch our use of the term seminal in relation to the talk that Tracy just gave, I think. So, but again, I also want to add my thanks and excitement about the science that we heard today. For me personally, it's a great day just to have a chance to sit back and to interact with and to listen to all of you. And I know to our trainees, it's a terrific opportunity as well. It's unfortunate that we couldn't all be together, but we're making the best of it. And I was aware too of how exciting it is, but how much other fun we have too when we get together. And hopefully next year we'll be able to do that. I can see we got the save the date sign here for next year. And Richie and I will shortly be thinking about who's going to be coming next year. We'll get that out to you soon. Yeah, want to again acknowledge our sponsors. Very much appreciate Corcept Therapeutics' ongoing support for this event. Another thing to mention is that when we do it live, we also actually have travel awards for trainees across the country, actually internationally, and give scholarships basically to bring up to 20 people here for the meeting, and Corcept has been very helpful with that. The Department of Psychiatry and Healthy Minds, as well as Health Motions Research Institute. And I think we're going to go to we've got the poster session coming up.
at 4:30.
At 4:30, and we hope that you all hang around for that. We'll participate. It's another, I think, innovative thing that's happened out of our virtual things that we can actually visit with the poster presenters and have conversations in each of the little rooms. So thank you again, all, and thanks for the attendees too for doing this and taking the time out of their day to be with us.
And thank you, Amber.
Yes, Amber, thank you. Okay, well, we'll hopefully see some of you at the poster session.
Very good.